Hi, everyone. Today I'm going to be talking about how brain support cells organize themselves and communicate with one another. Specifically, I'll be talking about one of the tests run by Dr. Baldwin's research team during their study to look at hippocamp's impact on astrocyte territory. Now, I know those terms might sound complicated, but don't worry. I'm going to break everything down so hopefully by the end of this, you learn something new. Before we talk about the specific test, let's learn what an astrocyte is. First, they are star-shaped cells in the brain with multiple arms reaching in all different directions. Second, they are support cells. They help brain cells communicate with one another. Third, each astrocyte claims its own territory in the brain. Think of it like it's having its own neighborhood. And fourth, astrocytes spread out their branches to fill that space or said neighborhood and reach the neurons that they need to support in the brain. Now that we know what astrocytes are, let's talk about hippocamp. Hippocamp is a molecule on the surface of astrocytes. Think of it like a connector protein. It helps cells recognize and interact with their neighboring cells. Without hippocamp, astrocytes can't properly communicate with other astrocytes next to them. Finally, hippocamp helps impact how cells develop their shape and claim territory on the brain's tissue. Now, let's talk about the study. So, what is the study about? Researchers wanted to see what would happen if they removed hippocamp and what would go wrong because of this. For this presentation, I'm focusing on one specific test they conducted. This test is when the researchers wanted to measure astrocyte territory formation. They wanted to know if hippocamp affects how much space astrocytes claim in the brain. The researchers used many methods in order to test astrocyte territory formation. During this experiment, mice were the main subjects tested on. First method they used was they injected fluorescent markers into young mice brains. Think of it like giving astrocytes a glow-in-the-dark label so that they could see them under a microscope. Next, they used microscopes to capture 3D images of astrocytes. Then came the measuring part. The researchers calculated how much space each astrocyte covered, mapping out the cell's territory volume. They did this for both astrocytes and astrocytes missing hippocamp. They also analyzed whether astrocytes were overlapping with their neighboring cells. Now let's look at what the researchers found after testing. The main finding here is that astrocytes without hippocamp had significantly smaller territories than those with hippocamp. The researchers also noticed that astrocytes with smaller territories had fewer branches. After comparing measurements tested, the difference was clear. Astrocytes without hippocamp claimed a lot less space than normal astrocytes. Here's a more simplified version of the results. On the left, it starts with astrocytes with less hippocamp. Then it goes to small territories as astrocytes can't claim their normal space in the brain anymore. This then leads into fewer branches being formed. Because their territories are smaller, the astrocytes are growing fewer branches. Finally, this changes the brain's signaling as astrocytes can't function the same. This is the figure used in the study to represent the findings from the specific testing. This figure shows the territory measurements and the different tests run. There are normal astrocytes on the left and the astrocytes missing hippocamp on the right. The red lines seen on the left bottom images are the lines that were used to map the territories on the astrocytes. Overall, the images show that less hippocamp means smaller territories. So, what's the big picture? Losing hippocamp disrupts how astrocytes claim territory and interact with neighbors. This matters because cell territory is critical for brain health. Without proper territories, astrocytes can't support enough brain connections. This is what can lead to neurological disorders like schizophrenia and why this research is so important to continue testing. So, while I know this seemed like a confusing topic at first, I hope you can leave this video knowing a little bit more on hippocamp or even learning what it was and its importance to astrocytes and brain development. Thank you so much for listening.